ബിജു സാറേ തുടങ്ങോ? അല്ല എന്താ വെയിറ്റ് ചെയ്യണോ?
എന്താ സാറിന്റെ അഭിപ്രായം? 10 സ്റ്റാർട്ട് ചെയ്യുന്ന അല്ലേ
സാറേ?
10.
ആ ശരി സാർ. എത്ര മണി സാറിന്റെ വാച്ചിൽ എപ്പോൾ എത്ര?
10:06 ഒന്ന് പറഞ്ഞേര്. അല്ലെ വേറെ എന്തെങ്കിലും സംസാരിക്കണോ സാറിന്?
വേണ്ട വേണ്ട, സംസാരിക്കാം, കുഴപ്പമില്ല. ആദ്യം ഒന്ന് കയറിക്കോട്ടെ. ഓക്കെ. Good morning all of you. The program will start by 10:10. Sorry for the inconvenience. Exactly by 10:10 the program will start. We'll start sharply by 10:10. So remaining participants yet to join. Just few more minutes. We'll wait and we'll start. Good morning, all of you.
Good morning.
സാറേ
അതെ അതെ തുടങ്ങല്ലേ?
പറയുന്നത് കേൾക്കുന്നുണ്ടോ?
കേൾക്കുന്നുണ്ട്.
സൗണ്ടിന് കുഴപ്പമൊന്നുമില്ലല്ലോ?
ഒരു കുഴപ്പമില്ല സാറേ. പ്രൊസീഡ്.
സ്റ്റാർട്ട് ചെയ്തു.
Okay. Good morning, all of you, and I welcome you all in behalf of Sidus International Publishing, Kerala, Palakkad, and day 2 today. So today's session will be delivered by Dr. Bijay Matthew sir. So I invite Dr. Bijay Matthew sir for deliver the talk. Before that, I just would like to tell you that I think you enjoyed the yesterday's session, and today during the session I request all the participants not to interfere with the session. After the session you will be having the half an hour time, 11 to 11:30. We are giving the discussion session. At the time you have to introduce yourself, then you can ask your questions whatever that, and Dr. Bijay Matthew sir will be clarifying your queries whatever it may be. Now this I would like to invite Dr. Bijay Matthew to deliver the talk. Sir, you can share the screen, sir. Screen share കൊടുത്തോളൂ sir. Vijay sir. Screen share. Yeah. Yes. You can proceed, sir. Sir, the audio just you. Audio.
Hello, good morning to all. Can you able to hear my voice?
Yes, sir.
Yes, sir.
Good morning. Yes, sir.
Okay, now you can
Very
mute
good
your.
morning, sir. Okay, sir.
Okay, okay, please mute your audio.
Okay.
Okay, today the section name is Medicinal Chemistry Research Lab and the artwork of Medcom research paper. So yesterday we discussed what are the criteria for publishing a paper and what are the documents what are the documents are necessary for submitting a paper and how to make an effective document. So all the things we showed yesterday itself. So inspired from that findings, today we are going to see how we can execute our research in a normal laboratory. So commonly people are thinking the research can be happened only in a research lab, something like IIT or CSIR or CDRI, like that. So you can start your research in your small laboratory itself. So I will give what are the tips or what are the common factors which is necessary to maintain an ideal medicinal chemistry research lab. So once you maintain your medicinal chemistry research lab, so then only you can start writing the paper. Okay. So you cannot generate the paper directly. Obviously, you before working on a paper, you should do the a basic research in your lab. So today we will see that how we can implement the basic research lab in our college itself. Okay, you see here, this is a simple documentation procedure which I followed in my lab. So what are the simple documents I can maintain in my medicinal chemistry research lab? It is not only for medicinal chemistry, all the disciplines which is related with the science you can keep like this. The first thing is keeping a laboratory notebook. So laboratory notebook, it's a personal notebook. So it should need the what is that the data which is we are going to the daily routine of data we are going to analyze in this particular area. So by doing the laboratory or keeping the laboratory notebook, so what are the necessities of laboratory notebook? The number one is we can observe a reaction with a specific time. So please note that when you are proceeding any reaction or any experiment in your lab, you please observe that particular reaction with a specified time. So always you please note that the date and time of that particular or corresponding work should be noted with a specific time. And what is the necessity of keeping this time in? So you can refer in the future. So while you are writing on the paperwork, so this time definitely it should not be, you will never there is no research paper need the time. But if you are getting more confident to write a paper, so the observation of the reaction and its time, this is a conventional type of thing, but you should follow this. And by keeping that particular time, so what is advantage means it will just prove which inventions you made exactly and what is the time. So the time and the nature of invention you can easily identify by keeping that particular date. So please note that the date is very important. And the next is if you're doing like this, sometimes we know that most of the, I think 90% of the participants are belongs from the academic background. So obviously, so Simon, we have to teach our students at the same time we should engage our uh, research. But if we are seeing these types of multiple tasks, obviously there may be a chance for the confusion. So to avoid that confusion only, we are keeping a laboratory notebook. So that is a last advantage. A researcher should write their idea. Suppose if you identify, I will tell a simple example in a chemical reaction. Suppose A plus B, the procedure is you are refluxing 5 hours in presence of a base or something like that. So during the first hour, you can see that. So there may be a chance for the formation of some slight color. After the 2 hour, you can see that it will disappear. You can see some other color. So if you are seeing the progress of the reaction, so each progress of the reaction we can easily identify by the observation. So you please note that suppose if you identified your product by means of TLC after 5 hours, so you please note that at that particular time, what color you got in that particular time. So you please note the time and the observation. Suppose if you're keeping such type of records, so that's why I told that in the last sentence, the researcher should write their ideas and maintain a file. It will wastage the time of rediscover the old ideas. Okay, so obviously you got an idea. So immediately, once you got an idea, immediately you please transfer your idea in your laboratory notebook. So that is very, very useful. Yesterday itself, I told that the heart of the research paper is discussion. During the discussion, so you can implement your idea which you gathered from your observation phase. So laboratory notebook is very important. The main purpose is yeah, the observation capacity can be refined, and it will just uh, leads to the wastage of rediscover your old ideas. These are the advantages of your laboratory notebook. Please keep in mind that. So in some of the labs, they will tell that logbook or laboratory notebook, both are same only. Okay, now so why we are going to make results, you see here, recording the results, that whether it is pharmacology results, whether it is Sutix based cognosty or chemistry, recording the results, it is one of the major essential what is that, the major essential part of your research. So especially, so this is a fundamental ethics: accurate reporting with no manipulation. So the last section itself, I told that, so no need to manipulate your results. Suppose if you got a bad result, so you cannot decide whether it is bad or good. The bad or good results always will judge by the audience or your world, only it will judge. So whatever the results you got during the time of your research, please report accurate results with no manipulation. Okay, so please keep in mind that. And another thing, that suppose if you are reporting the exact procedure, so it will clear that, I think last slide itself I told that, suppose if any new person without prior of the knowledge, so he can easily repeat the experiment. Suppose if a principal investigator, so if he is giving some work to the student, so obviously if you are giving the exact procedure and all the observation phases, so he can follow the experiment, or he or she can follow the experiment, so he can easily repeat the entire experiment without the prior knowledge. Suppose if you have the basic knowledge of handling the chemicals, so he can easily reproduce the results by uh, seeing the laboratory notebook. And please note that always in your synthesis, please note the melting point of your corresponding compound before and after recrystallization. So that is very important. So but in case of your what is that, in the publication process, they will only consider your melting point of your compound after the recrystallization. But anyway, this change in the melting point pattern, so you can note that whether the crude product melting point and after the recrystallization, so by comparing this, you can easily identify whether there is a presence of impurities or they are not. Okay, so please note that melting point should be noted before and after the recrystallization, but only you can note the melting point after the, only you should, it should be in, only you can report in your paper. And the solubility pattern, that is very important, because solubility pattern is one of the essential criteria. So it will also have a high significance for getting the exact solvent for your biological activity. At the same time, TLC pattern for the separation of molecule via column chromatography, all these types of experiment. So we need the solubility pattern of your molecule. Okay, so that's why during the time of your synthesis, you please keep your all the solvents. So all the solvents means from polar to non-polar, something like water, ethanol, methanol, acetic acid, water, ethyl acetate, benzene, toluene, all the these type of polar to non-polar, chloroform, benzene, all the things you can maintain your lab. So it will just, you please check the solubility pattern of your compound, whether it is soluble or insoluble or partially soluble. So these the nature, it is very very important, the solubility pattern of your molecule. And please note that the yield and duration of action, duration of reaction is also very very important. And the next one is the recording results. It is TLC of the solvent phase. That means, all you know that TLC, it is majorly we are focusing on the completion of the reaction. Suppose if you got a single spot which is entirely different from the starting material, you can identify the formation of a new compound. So please note the TLC phase. Suppose if you are mentioning the TLC phase in your research paper, if a new person will try the same suppose if he is failing in that particular solvent phase, so it will just lose the credibility of your work. Please aware about the TLC of your solvent phase. And this is a very important thing. Unexpected happenings should be clearly recorded at the time. This is a clear factor. So suppose if you are seeing, we are following a particular reaction on the base of a literature. So if you are following the basis of, if you are following a literature based study, sometimes there may be a chance, it depends upon, you know that the chemistry and between the chemical reaction, it may be varies, depends upon the nature of your starting material. Suppose if there is an unexpected happening, something like, suppose if you are stirring or refluxing the molecule, so if after 1 hour or 2 hours, so if your product has a tendency to evaporate, or if your product has a tendency to stick on the walls of your reaction vessel, so sometimes that type of unexpected happening, commonly you cannot see in your research paper. Obviously your literature review, by seeing the literature review, obviously they will omit this type of unexpected happening. Suppose if you are receiving such type of unexpected happening, so you please should note that, so that you can record it with the corresponding time. So this is very important, that is no manipulation. And I told that what are the other parameter which is needed to be recorded in your laboratory notebook. And this is the another major problem we are suffering. You see here, I think the last session itself, I told that, so the difference between a scientific writer and a normal writer. Okay, so normal writer, so I can write a poem, I can write a story, a short story, so anything I can write. But if you are if you are a scientific writer, so we should face the problem, because whenever, if you're seeing a normal writer or short story writer or poem writer, so he can execute the ideas on its own way, his own way. So there is no limitation, there is no boundary in that. So he can execute or she can execute in his own way or her own way. But in case of your scientific writing, so we cannot do like that. All the discussion or all the things we should be maintained on the basis of the basic concept of science. So that's why I just classified into two. You see here, suppose the difference between the medicinal chemistry research and a normal clinical research. Okay, you see here, in case we have in-depth knowledge of drug candidates. So if a medicinal chemistry people, if he is doing a particular research on his drug design project. So maybe he have 100% of knowledge about the structure and what type of mechanism you can expect. All the things he should know that. But you please see here. Suppose if you're going for a pharmacy, you know that pharmacy, I think they were our ultimate profession is pharmacy or working as a pharmacist. So in that particular area, so you no need to explain the structure active relationship, you no need to explain the basic ring, so nothing, we are just giving what is that the in what is that only the indications of your drug. So that means that is a difference. And the next is the medical chemistry research, the audience is very small, and in other cases the audience is very large. And you see here, and if you're going for a research, it should be applicable only in professionals, but in the other case, so anyone can be go, suppose if you have a basic degree of pharmacy, so he can do that. So that is a difference between the real problem we are facing. So our audience is very small and strong. Okay, please keep in mind. And I can tell that this is my point of view. If you are considering medical chemistry research, they are the real architects of a drug. You know that without structure, so you cannot imagine a drug molecule. So that's why if you're considering, the real architect belongs to the medical chemistry field. And you see here, they are living in the different field, and they will, oh, you see here, if you're going for a medical chemistry research, you can see that always your mind have huge amount of mechanism. So obviously they will never have a thought of smile. And these are the, and always they will never compromise the quality. These are the common things. This is the attitude of medicinal chemistry researchers. You see here, the medicinal chemistry, you know that it's a combination of both organic chemistry and molecular pharmacology, which includes biochemistry also. So attitude of the medicinal chemistry people, which is entirely different from other areas, and it is an endless world of a jungle which connect with the biological significance of the molecule. So please note that you are very unique in this field. Okay, so there may be an opposition I can expect during the time of discussion. Okay, we will see in that. Now you see here, this is the real medicinal chemistry people. You see here, many of the peoples, they are doing the research, they have their own pathway, you see here. But you can see that so we are unique, that's why we are going in a different pathway for selecting the molecules and executing all the things which is connecting with that. So uh, our path is entirely different. Okay, now you see here, this is a professionally oriented medical chemistry research lab. You see here, I think that guy is doing the separation by column chromatography. You see here, so the he's we are just entering to the world of our lab. So for doing the what is that, our chemical experiment, synthesis, purification, all the things. Okay, now we will see that this is one of the most important part of your research. Suppose I will tell a simple story. If you are synthesizing any molecules in your lab, the purification is one of the major problem. You know that, suppose if you are doing multi-step, something like multi-step or three-step or four-step process, so problems of purification is a common thing we can arise during the time of your synthesis. So at that particular time, so we will use some tricks which is basically used to resolve this type of problems. The first one is washing with water and washing with a sufficient amount of solvent. Suppose if you are uh, after the formation of your product in your whether it is after the filtration, so you please wash with your product thoroughly with water. Suppose what is the use of doing these things by washing with water. So suppose if any acid impurities or alcohol impurities or basic impurities which is present in your crude products, so maybe 50 to 60 percentage of impurities can be easily eradicated by washing the water. So please note that the washing water is one of the very important. So one after the washing only, you please go for your drying process. So once you are drying in a proper manner, and now you are going to make another most important purification technique, that is recrystallization. So that name itself, we know the meaning. Recrystallization, re means again crystal. You are making the crude product into a crystalline format. We know that crystal is a stable conformation, so there is no issue in that. You will get an uniform crystal lattice. So obviously you can expect a single compound in that. Okay, so most of the time recrystallization is very important. And now the next slide, we will see that what are the common parameter we are just focusing on recrystallization. Very important. Please note for the medicinal chemistry people who are working on that, while doing the recrystallization, you will get always a well organized rigid structure. Okay, I told that in suppose if you are identify, there are two forms of compound: amorphous form and crystalline forms. Suppose if you are getting a crystalline form, you can 100% thoroughly in a confidence of organized crystal lattice. So during the time of crystallization, so you please note that, I think, can you see the third point is very important. It should not be soluble or partially soluble in cold condition. Please, that's why I told that during the time of your synthesis, you please keep all the category of solvents from polar to non-polar. Okay, so in the initial time itself, you please check whether your compound, I already told you, already we know a basic concept. Polar substance can be solubilized easily in polar solvent, and non-polar compound can be easily in non-polar solvent. Suppose if you are seeing the combination of both polar and non-polar compounds, so solvents, depends upon the ratio it will be solubilized. So solubility pattern, identification of the solubility pattern is one of the very important in both TLC and recrystallization pattern. So you please note that your solubility is one of the major important factor, and your substance should not be insoluble. Sorry, substance should be insoluble or partially insoluble. Okay, in the cold condition, but it should be soluble in the hot condition. So all the, if your compound is soluble in the hot condition, it can recover its original state, that it can recover its original well organized crystal structure after cooling. So that's why, so this is the recrystallization. And the last is very important: the slower rate of cooling will get the bigger chance of crystal. That I think commonly, so in our people, what we are doing means, after the hot condition, immediately we will dip into the ice bottle or ice bath. So in some compound you will get, there is no issue in that. So but my point of view, if you are making, that means if you cool your reaction hot mixture in a normal temperature itself, you know that if you are cooling the in normal temperature, the cooling rate will be slower, and there may be a good chance for the well organized crystal. Okay, so you please keep in mind that if you're going for some big synthesis, so don't incorporate your substance or reaction vessel directly into the ice. So in some cases you will get, but in most of the cases, the exact theory is, if you are slowing the rate of cooling, the chance of crystallization is very high, and you will get very nice crystals. Okay, please, this is these are the important things you should know during the time of your recrystallization. Okay, now I got, you see here, so maximum, I think hello.
Sir, there is a lot of disturbance.
Hello. Hello, can you able to hear?
Sir, yes, sir, but little bit disturbance, sir.
Disturbance.
Sound is coming, sir. Someone is hitting something.
Hello.
Hello.
Hello, Biju sir.
Now it is okay, sir.
Okay, okay, fine, fine. Biju sir. Biju.
I think sir mic was muted.
Hello.
Okay, okay. Okay, sir.
Okay.
സംസാരിച്ചോ 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 സാറേ ബിജു സാറേ
Okay, okay.
ഓക്കെ ഫൈൻ ഫൈൻ സൗണ്ട് കേൾക്കുന്നില്ല കേട്ടോ
Okay. Now, can you able to hear?
യെസ് 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 യാ
Okay. Okay. Once you got some results, you see here in a normal synthetic laboratory, these are the maximum thing you can do. That you can synthesize a molecule and you can purify. That's all. And you can identify the physical characterization, something like melting point and TLC. That's all. Okay. Once you got some good molecules in your lab, then your aim is you are dreaming the project. Okay, I think yesterday itself I told can you able to hear clearly now?
Yes,
Yes,
sir.
sir. Yes, sir.
Yes, sir. Yes, sir. Yes, sir.
Okay, okay, now your aim is you are dreaming your project, and after the biological evaluation, also after the completion of the project, you see here, can you see the next is you are going to write a paper. So writing a paper, yesterday itself I told that what are the hurdles we are commonly facing, and submit a peer reviewed journal. And the next step is we are waiting for the fate of your work. Finally, you got the decision from the journal: accept, revise and accept, resubmit. The last one is terrific. Can you see the last one? It is reject. So I think the first decision, I think 99.9% of journal, it will never accept any of the paper without revision. So obviously, if it is accept directly, maybe the submitting author is an emeritus professor. So commonly in all the journals, you can expect this decision: revise and resubmit. Sorry, revise. Once you are going the revise, if it is satisfied by the editor in charge and your reviewers, it there is a chance for the acceptance. And the next I saw, can you see it is, I just mentioned it is. Hello. Next is you are going to the next is resubmit. Resubmit means, suppose if any reviewers will oppose your work, sometimes the editor will give a chance to resubmit the same work in that same journal with a new manuscript number. Okay, so at that particular time, you can expect maybe little bit hope it's there is begin in that, because resubmission, it is typically depends upon the decision of editor in charge, because the editor in charge have some interest on your paper, so he will give another chance for resubmission. So if you are going to resubmit, actually in the last year 2018, so one of my paper in Royal Society of Chemistry, so that is medcom communication, I think I will show you in the during the time of discussion part, I will show you. So I think comparing by evaluating 4 reviewers, 3 reviewers accepted and give the revision, and 1 reviewer is rejected. Normally Royal Society of Chemistry journals, it will never, if any reviewer will oppose the paper, they will never give a chance for the resubmission. But in my case, he gave a chance to resubmit the problems which arise by the reviewers, that is the reviewer who rejected my work, and I submitted the work. And this is, I can tell that this is a simple B Farm work. That is a interesting thing only. It is accepted finally. So that's why resubmit is another chance. And the last one is, we are commonly facing this one. What is that? Reject. Once you are rejecting a paper, once your paper is rejected by a journal, either by editor in charge or peer reviewer, please don't worry about that. Find a new journal. Okay, don't think that rejection of paper is the ultimate fate of your work. Rejection will give another in my point of view. Rejection, it is meaning the another point of your work. It will give an another venue of your work. It will give an another chance of your work with improved finding. So don't my request is, dear participant, suppose if you received any rejection letter, because daily I think in monthly I will receive more than 10 to 15 rejection paper. So then only you can see one or two paper will be accepted. So rejection, it is not the ultimate thing. You please refine your work and try to submit a new journal, or also you can try to submit the same journal with improved finding. So that is one of the very important thing. And now this is the idea is okay. Now the next is, yesterday itself I told that the how to write an abstract. Now these basic concept I am just implementing my medicinal chemistry paper. Okay, please note that if you're seeing a medicinal chemistry paper, we have a problems. Okay, suppose your molecule is already synthesized or not synthesized. That means if it is new or reported. I think sometimes your molecules may be reported, or sometimes your molecules may be new. So you please keep in mind that you should know all the history about your molecules which you are going to synthesize. Suppose if you have a molecule in your hand, you should have 100 confidence whether it is novel or old one. So if it will old one or new one, there is no issue in that. You please, in the abstract, can you see the first point? It should be mentioned, if it is novel, you should mention newly synthesized. Okay. Or another option is you can mention novel molecules, something like novel typhin based candidates. So these are the common terms you can employ. So if your molecules, please note that if your molecule is reported, don't write this. Obviously the reviewer will reject directly. So you please keep in mind that. So that also I already told that in the last time itself, in the cover letter, you please mention all the history of your molecule. The second is, suppose if your molecule is not novel, no need to worry about that. You start your abstract with, what is that? It is previously reported. Okay, oh sorry, I just mentioned the duplicated, I made the same. Wait, sorry for that. Previously reported. Okay, you should mention it is previously a reported molecule, or previously identified molecule, or previously synthesized molecule. So previously documented molecule, any terms you can use. And the third time, can you see that 12 molecules of pyrazolin? My dear participant, don't start with in an abstract with a number. This is one of the fundamental concepts. So always, that's why I mentioned in a red line, you please don't start with the what is that 12 molecules. You always, the beginning of the sentence, you please don't start with the number. Okay. And always you please note that you can use the lead molecule and the code and IUPC. IUPC is very, very important because your molecules details are appeared in PubMed Index and Scopus and Web of Science. All these international agencies, so they don't want the code something like 5A or TA. So you please code your IUPC. IUPC nowadays it is very easy, because if you have a ChemSketch software, you can generate any of the molecule IUPC. Okay, so there is no issue in that. And the next is, always in your abstract, you should mention the IC50 value, Ka value, effective drug concentration, and MIC. Okay, that means the results and discussion, the what is that, the nature, that is whether it is you are representing the molecule biological activity in a way of IC50 or in a way of inhibitory constant or way of ED50. All these parameter is necessary. And this is another interesting parameter. No need to make any spectral interpretation of your molecule. That's, there is a separate part is present in the manuscript, so you can concentrate on that. Please, in the abstract, no need to do the spectral interpretation. And please note that I think the last itself I told that you should make a clear hypothesis and ensure that your science is true. Because why I'm telling that? So you see here already, I think, can you see that in the abstracts, these are the major things you should know: that aim, objective, materials method, results, conclusion, it should be in a concise format. Okay, so in another way, I can tell that a scientific writing abstract or the scientific article abstract, I can tell that it is the entire story of a author or entire story of whole research. He is expressing in a minimum words with the variety of audience or maximum audience, because by seeing this abstract itself, the audience should attract towards you, attracts towards you or your work. So that's why if you are seeing the entire research paper, all the research paper, you should concise format, make the aim, objective, materials method, results, conclusion, and the whole the entire story with a minimal weight, with the maximum audience. So the attraction of audience is the prime aim of a research writing paper. So this is the because already I think there is no need to mention the tense. Already I told that in the last session itself, what are the things you should needed for the abstract. So this is a typical medicinal chemistry paper. Don't use if it is novel, you can use newly or novel. If it is reported, you should not reported. Don't start with the number. Always use IUPC. You should mention the biological significance in terms of IC50 Ka. No need of interpretation. Hypothesis must be very, very clear, and it should ensure that it should within the science. And the next is the introduction. You know that I told that yesterday itself, what is the common whether it is medicinal chemistry or pharmacology introduction, it is nothing but the basic history of your work and how we are going to execute, how we are what are the previous literature connecting with the history, and what are the gaps present in the history. Now we are just focusing on my medicinal chemistry paper. The first paragraph, please note that the pathogenesis of disease. Okay, in medicinal chemistry, we know that we are designing a drug molecule for targeting a particular disease. So for targeting a particular disease, the in the introduction itself, the audience should have 100% awareness of the disease, whether it may be old disease or new disease. There is no issue in that, but you should make a detailed knowledge about the pathogenesis of this disease. And the second point, you should note that selection of the target. Suppose in a disease, there are, suppose if you are going to make a drug design of cancer, you know that in the cancer there are many targets are there: cytokinase, cyclin dependent kinase, tyrosinase, topoisomerase. So many targets are there. So the selection of target is one of the very important, that so you identify the disease, give a detailed description, and then you please slowly land it to the selection of the target. Please note that all these things, you please take the latest discovery of literature. Suppose if you are going for the pathogenesis of disease, commonly you please note that you please refer clinical based journal, something like Lancet, Medical Hypothesis, a New England Journal of Medicine. So if you are referring that particular type of clinical based journal, you will get a detailed knowledge of pathogenesis. Please, in the introduction part, you please concentrate on these journals to gather your literature. And please note all the latest 5-year discovery. Suppose if you are taking all the discovery, you know that it is, we cannot make all the things in your abstract, sorry, in your introduction. So at least you please refer the latest cutting edge of your theory, sorry, your literature. And you please note that the main finding of the literature, you please mention in your laboratory notebook, and you can keep in that. And the second part is very important. Once you identified the disease, selection, and discovery from the latest journals, then your aim is how I can design a molecules on the basis of this target. So there are many nucleus are present in our world. Many heterocyclic nucleus are there. Many aromatic, heteroaromatic, what is that? Aliphatic nucleus are there. So the next your aim is we should focus on the chemistry of the nucleus. So what type of the basic ring or what type of molecule you are going to explore in your introduction, you please make the detailed description of your nucleus. So please keep in mind that in that area, you please refer the European Journal of Medicinal Chemistry, Bioorganic Chemistry, in mini reviews in organic chemistry, chemical reviews. These are the journal. You will get the exact chemistry of the nucleus. So please refer that typical journal. You will get a detailed knowledge about the chemistry which we are going to explore. And the next is this is another. I told that yesterday itself. Whenever if you're making an introduction, the introduction should be very attractive. Like not only in the introduction, the whole the paper should be attractive to the audience. Okay, so that's why. Suppose if you are taking the structure of indole. Okay, if you're taking the structure of indole, we are going to explore some drug design concept which is connecting with the indole. So you please note that what are the approved drugs with the indole nucleus in this particular target. Make sure with the diagram. Suppose your indole nucleus, maybe you know that very famous anti-inflammatory indomethacin is there, that are common FDA approved drug. So you please, I think if you're going for the drug bank, what is it? Details. You will get the detail about the FDA approved latest drug molecule and its nucleus and its target. So you please quote your nucleus with the target and please make sure exact diagram. So that diagram also you can implement your introduction. It will give some colorful manner in your introduction. Please note that. And the previous history of the molecules, it is you can make in a SAR. You know that SAR is a very common term we can see in a medicinal chemistry life, that is structure active relationship. So you please quote all the structure active relationship and please mention in your introduction by means of some graphs, sorry, by means of some diagrams. I will show you that how you can make that in the remaining slides. And the next is, I think, the very common fact. You identified the disease, you identified the target, you got the literature from well-established clinical journal, you got the literature from the well-established nucleus, you found the nucleus, you designed the nucleus, you identified is there any nucleus is belongs to the target, and you identified the history by means of SAR. All these things will give the gap which is present in the work. Okay, now by gathering all these work, you can easily identify that what is a find of the gap. Okay, now you please note that how your synthetic scheme can expand the SIR. This is the most important part in your introduction, because many reviewers will oppose, because I think commonly you can see a decision from the reviewers. There is no novelty in the work. So that's why I'm just going to reject the paper, because you please keep in mind that whenever if you're exploring any chemistry related paper, this particular area is very, very important. You please convince your reviewer or audience this is the gap. On the basis of that gap, I can already, there is an established structure active relationship is there. This structure active relationship I can expand is nothing but I can enrich more amount of principles of medicinal chemistry on the basis of my synthetic scheme. So you please give a clear idea about the synthetic scheme can expand the SIR. Please note that this is one of the major important. And please clearly mention your objective at the last paragraph. So your last paragraph will tell that what are the things we are going to explore. You already identified the gap. You ensure your audience this in the scheme can be expand the work, and on the basis of the expansion, these are my objects. So I think I told a very systematic manner. Are you okay? Hello.
It's excellent, sir.
Yes, sir. Yes,
Okay.
sir. You can.
Okay. So this is a way pathogenesis, selection, latest discovery, literature, chemistry, and approved drugs, history. You see here, you can, because this is my way of format, you can also change. There is no issue in that. So this is a common format, or this is a common attractive way of making the introduction with more beautiful manner. Okay. Now this is the, I think last year my publication in the Journal of Enzyme Medicinal Inhibition from the Taylor and Francis, I think it is published in 2020. We worked this particular molecule. It is a collaboration work with a Turkish lab and Italian lab. Okay, so we designed a molecule. So basically the molecule structure and its basic scaffold is old. Can you see that? It's a very complicated molecule. You see here, you can see a benzene, a linker, a pyridine molecule, another linker, again a benzene nucleus, very complicated molecule. You see here, by identifying the literature, I just made the structure active relationships. Can you see that? Suppose if it is no linker, previously it is reported as analgesic, anti-inflammatory, and antimicrobial. Suppose if your linker is, what is that? If it is a pyrazine, it will show us the analgesic, antimicrobial, anticholinesterase, and finally you can see that an anti-cancer activity. So that means this is a core concept. So anti-cancer activity I got because, so these structure active relationship you cannot see in your literature. By gathering the literature, you are making a new structure active relationship of your molecule. That means structure active relationship, in a simple way I can tell that it is the history of your work. Some eminent scientist did the work and you are correlating, or you are making, suppose if you know this particular diagram, the reviewer can easily identify that, oh, the particular man is going to synthesize or exploring this particular area, so he can easily tackle the your literature easily. So that's why, so this type of graphical representation is very necessary. It will just explode. Suppose in the last, can you see that the R is 2 fluorine, 4 chlorine, and the R is there are two substitution is there. The first, there are two benzene rings are there. So that's why I just distinguish by means of R and R1. So you can see that some substitutions are there. Suppose if you are introducing a new substitution, obviously that is a novel work only. So that type of implementation, that type of alteration you can do in the existing scaffold itself. The concept is, if it is old, there is no issue, don't worry. If it is previously reported, and don't worry, your concept should be novel. Okay. Next is, this is the, most of the people are suffering this particular area. You see here, the spectral interpretation. Okay. You know that if you are writing a medicinal chemistry research paper, I know that I think many people from the ISF College of Pharmacy, many good colleges are there who are actively participating in the medicinal chemistry research. I can frequently see in my Facebook pages, they are synthesizing many molecules, many characterization, very JSS college. I think very good colleges are there for actively synthesizing the molecules. And this is one of the most important part, the spectral interpretation. So you please keep in mind that if you are a medicinal chemistry researcher, first thing is you should have a thorough knowledge in the organic chemistry. You should have a thorough knowledge in the biochemistry, and you should have a thorough knowledge in the interpretation of the molecule. Okay, this is the most typical aspect, interpretation. Suppose if you are unable to interpret your molecule in a successful way, you cannot make in the category of chemist. So chemist is, he should be able to interpret your molecule by seeing the NMR and C13 or mass spectroscopy, at least by the proposed structure. Okay, I cannot tell that unknown compound it is, that is also very difficult one. But anyway, he should know at least this particular concept. And he should also know about the computational chemistry also. That means medicinal chemistry is an interdisciplinary subject of all these things. Now can you see that what type of things I made? That can you see that in the first materials and methods, I already told you know that what type of tensors we are using. Now we will see that procedure of the synthesis and physical characterization and spectral. Please not always you please mention the code, and code should be always bold. and then what is that yield, and then the yield is my means of percentage. Now the HNMR, can you see that it is 500 megahertz? So that is depends upon, you can use the 200 megahertz. So why we are mentioning 500 megahertz or 200 megahertz? You know that the basic answer. Suppose if you want to get a multiplet peak, some reviewers they will ask the coupling constant. So at that particular time, you please differentiate the coupling constant and just multiply into your corresponding megahertz which is using for your NMR, and you will get the coupling one. So for that purpose only, I'm just mentioning the, because some of the reviewers, if you are very strong in the chemistry, so obviously they will ask the coupling constant. If it is singlet, there is no problem in that, because here I didn't mention about the coupling constant. I just simply mentioned about the range between the 7.541 to the 7.31. I just mentioned it is like that. Okay, now I will show the you see here, actually yesterday I planned for the C13 and mass spectroscopy. Unfortunately, I think we have very limited period of time, so that's why can you able to skip, or can I continue the slide?
Continue. Sir.
Do you want to interpret this molecule? Do you want to see the interpretation of the molecule?
Yes,
Yes,
sir.
sir.
Do you want okay. This is a very simple molecule, which is, you can tell that it is a methyl thiosemi carbazone, which is obtained in our lab, and it is published. It is an old molecule, but it is, I think, the last month we published in the Process of Biochemistry and Elsevier journal. Okay, you see that how the interpretation can be seen in that. You know that HNMR, we know that it is a typical interpretation which is connecting with the nature of number of protons. So here you can see that how many types of proton. A methyl proton is there, and a NH proton which is nearer to the CS, and a, what is that, another NH group is there, and a CH group, and a phenyl system is there. 1, 2, 3, 4 and 5. So these type of I think 5 to 6 types of protons are there. So you please note that CH3 group, can you see the first CH3 group? You please see. So in the graph first, can you see that that is a most shielded proton? You can see that it is present in 3.27. Can you see that in the integration value is 2.8? That means it contains 3 protons. So this part is okay. Now the next part is NH group. So you can see, now you please totally count the number of hydrogen atom. In benzene it is 5. 5 plus 1, 6, 7, 8, 11. Once again I will count. 3, 4, 5, 6, 11. You please count here. 1, 2. Here it is 2. That is 1, 2, 4, 5, 7. Sorry, it is 2.7. That is 3 only. So 1, 2, 3, 4, 4 plus 3, it is 7, and you will get the number of protons exactly. Okay, so this is a normal thing, and you can see that normal how many protons you can easily identify from the shielded proton. And because that is a because if I will going to explain, so definitely it will deviate the concept. That's why I just wrote that. Now we will see that how the shielding and deshielding protons can be placed in your. So you please note that always the shielded proton should be in the first. That is 3.7. In bracket you please mention it is appear in singlet. Number of proton is 3 hydrogen, and the representative group is CH3. You please note that is very important. And the second one is your multiplet, and the last one is, can you see the last one is singlet? That is the NH group which is nearer to the thiocarbamyl group. That is the most deshielded part which is away from the, that is, you can see that it is almost 9.1. So from shielded to the deshielded, you please mention that. And the underline, you please always mention it should be the representative molecule which you are going to explore. Okay, so I think in another seminar I will take that detail about how to take the NMR detail, because I think very limited time. So if you are going to explore, I think you may get bored. Okay. Now this is a mass spectroscopy. Please forget about this. Is the results. Already I told you now the results is mentioned by means of, you see here, you can see the results. Yesterday itself I told you know that this is one of the common results which is obtained from my lab, which is the recent publications. Okay, you can see that if it is plural, you can use the what is that, a simple present tense. So you can use that, and the plot. And this is a discussion. discussion. I told that this is the most important part and the heart of your part. And you just see that I just mentioned one of the introduction. You please that introduction of electron donating group methyl group at the para position of the phenol resulted in a selective inhibition in low concentration with a selectivity index of 78. Okay, suppose this is please note that I will tell some tips for the structure active relationship. If you want to explore the structure active relationship, you please keep in mind that the first one is you should categorize your groups into electron donating group and electron withdrawing group. That is what is that electron donating group? I think that is a fundamental only something like hydroxyl group, electron withdrawing group something like NO2 group. So in that electron donating group itself, again you please categorize into weak electron donating group, moderate and strong. And again your electron withdrawing group, you can make the electron withdrawing group at the same time lipophilic. What is that group? Can you tell that? Commonly the electron withdrawing group is, it is nitro group. Can you tell any more electron withdrawing group? Please, can you tell any more? The persons who are coming from the, can you tell any more, please? Now, hello, can you able to hear?
Amine.
Hello, can you
Yes,
tell
sir.
that
Yes, sir. Yes, sir. Yep. Yes, sir.
audience, can you tell that a nitro group is a common electron negative group? Can you tell any more electron withdrawing group?
All the halides are chloride and bromide and
Who, yeah, cyanide. Who told the hydroxyl group? Hydroxyl group is a electron donating.
Electron donating.
Electron donating group. In the halogen itself, please unmute. In the halogen itself, halogen is an electron withdrawing group. At the same time, in the structure active relationship, you can tell that it is a lipophilic group. So it is a same time it is an electro withdrawing group, but it can impart a more hydrophobic interaction. So electron donating group and electron withdrawing group people is categorized and its nature is categorized. Second thing is the position of electrophile. You know that the position of electron donating and electron withdrawing group. So whether it is ortho, whether it is meta, whether it is para, how this orientation will influence its activity. And now the next structural relationship commonly is the size of the ring, expansion of the ring, and the linker shortening, whether it is increasing the length or decreasing the length. So that is typically depends upon your synthetic scheme only. So commonly, if you are explaining all these type of structural relationship, you can establish the result on the basis of this. Clear? Are you clear? Anyone?
Yes, sir.
Okay. Now we will see. This is the last paper. I told that this is the last paper. I think you can see many authors. I think majority of the authors are from my lab, and many people participated, especially my B.Pharm students who participated for the synthesis. Okay, this is a very interesting paper in the Royal Society of Chemistry. I told that in the beginning itself, this particular paper was rejected by the one of the reviewer. He told that the drug design which I implemented is wrong. But the other three reviewers are accepted, but editor in charge again give a chance to hypothesis. Now, can you see that? Can you see two structures? Can you see?
Yes, sir. Yes, sir. Yes, sir.
So in the left hand side, that is a BEST inhibitor, SB11. That is a BEST molecule, and this molecule I just compared with my standard. But by comparing with the standard, my molecule is not that much of active. I already told that I got very moderate activity only. So I didn't get some potent molecule. I don't have any potency because I synthesize some good molecules, but that molecules doesn't have a prominent activity or promising activity when you're comparing with the lazibamide. Lazibamide, you know that it is a very famous FDA approved MAO-B inhibitor. So then the reviewer asked a question. You synthesized and designed the molecule, but unfortunately, so what is the use in that? Then I just make a correlation. Can you see the correlation? I just make a resemblance with my FDA approved drug. So in the FDA approved drug, can you see that the first portion is halogenated aryl system. In the FDA approved drug, you can see the halogenated heteroaryl. That means almost similar. I cannot tell that same. There is a similarity you can expect. The next part is, can you see that side chain? Here my molecule is 5 side chain with the 5 atom. That is 1, 2, 3, 4, 5 atom is present. Here the side chain which consists of 6 atom. That means length also may be same. Here I can tell that the terminal position is NH2 group. Here the terminal position is NH2 group. So I established a clear hypothesis which is connecting with my molecule. So this particular concept is accepted by the editor in charge, and finally the paper is accepted. So that's why I told that don't think about the results. Suppose if you got a bad result also, the interpretation which you are going to make a convenient way in front of the audience, that is a most acceptable one. So I just make a a comparison, and we can make a common rule. Similar molecule will recognize similar receptor. And I also made another advantage. You see here, if you're seeing this molecule, the molecular weight is less than 200. You know that molecular weight is less than 200 means it is obviously it will obey the Lipinski rule of Y. Always it have a good pharmacokinetic profile. So all these parameter which I implemented in the manuscript forced the editor in charge to publish, because I made a clear hypothesis on the basis of a scientific background. So he have no option to obey the third reviewer. He told that on what basis the third reviewer rejected means the third reviewer rejected because my molecule is not potent. But I can make that in the molecule is not potent, but this molecule can make a similar type of drug design concept of approved drug. So if you are modeling or if you are refining this structure in future, you can expect, suppose if the side chain if I will increase from 5 to 6 carbon, it can be increases. So such type of hypothesis, such type of scientific valid hypothesis, you can accept. You can implement your paper. So this is the latest paper which we published in the last year in the bioorganic chemistry. Okay, so there are many papers are there. So I just quoted 1 or 2 paper only. Okay, so this is a very clear structure-active relationship. Okay, we synthesized more than 26 molecules in my lab, and we compared the monoamine oxidase enzyme. Here, can you see that? Or can you able to see that the first part I mentioned the structural relationship by means of n. When there are n is 1 and 2, that means n, if it is 2, what is that? Ring is expanded. If n is 1, ring is 5 membered. If n is 2, n is 6 membered. So if the ring is expanded, I make a conclusion: the MAO-B inhibition will be increases. And also I make another conclusion for Br chlorine, that is fluorine, bromine, chlorine. What is the meaning of this representation? That means if you are introducing fluorine, high activity. If you are introducing bromine, you will get again the activity will become decreases. If you are introducing chlorine, actually when it is compared with the fluorine, it is less active. I think we wrote another paper, I think yesterday itself, that is published. What is the reason behind it? That we will see in another occasion. I will show that by means of molecular dynamics mechanism how this concept is true. Okay. And also I just maintained if RCA2H5 is ethyl group with the A, you can see MAO A inhibition, but all the other structure active relationship is concentrated on the MAO B. But if you are introducing a single group, you see here a total shift in the activity. So these are some interesting structure active relationship you can incorporate your paper. So depending upon the your ideas only, the paper will be accepted. Okay, so writing a paper and doing the sincerity in your research is both are very, very important. Okay. So this is my latest paper on the current medicinal chemistry. So that is a Q1 journal in the Bentham Science. I just mentioned what is the orientation of halogens. I think by seeing the graphical representation itself, we can identify that what type of structure relationship I introduced for this molecule. Sorry, the time is almost over. Okay, I can. So this is the latest paper in the process of biochemistry. Here also I just mentioned a detailed manner of how to make the structure active relationship by doing various groups of inhibition, by introducing the fluorine, by introducing the methyl group. You can see that. I think if I will explain detailedly, it will not I think the time is not sufficient. Okay. And conclusion, it is a clear statement and summary of the work. You see here, always you should mention the best feature of the lead molecule. Okay, so by seeing this discussion itself, you see here this discussion, if a reviewer will see this discussion, obviously he have a thinking that this particular man or this particular scientist who is actively involved in the subject. So that's why my simple request is, suppose if you want to do the PhD, don't do the PhD for getting the position. Okay, so some people are registering the PhD for getting a position from the lecturer to the assistant, assistant to the associate, associate to the professor. No, so you see here that type of PhD. So you can take that doctoral degree, but it is I cannot accept, because getting simple degree, it is not a conventional. Everyone can take the degree nowadays. Taking degree is not a what is a difficult task. But the question is, after your PhD or after your research or after your job, so what is the new things, what is the cutting edge you can incorporate your particular field. Okay, so that is very important. Okay, so you please go through detail. I think some of them have very interest on the medicinal chemistry. Okay, so if you have interest on your own area, you please expand your structure relationship. You make the best hypothesis, you make the best molecule. Sometimes you see sometimes it may be reflected in your future. Okay, so don't think that, okay, actually what our people we are thinking is that we suppose I will, or if I will tell a particular student or a teacher, can you do the synthesis, so he will tell or she will tell many stories. Sir, I have a huge amount of workload in my college because my principal is not allowing, and I have to write the what is it, work schedule. So many things you can tell that, so many excuses you can tell that. Okay, so I will tell a very simple concept. If any person, if you will spare at least 1 or 2 hours, I think monthly he can publish one review article. Dear participant, if you are sparing a 2 hour in a day for writing the literature, or reading the literature and writing the findings from that, you can, I can assure you, you can. But the basic things, you should have an interest. It is not a what is it, the process, it is not coming from instantaneously. It is what is it, it is, if you are practicing this particularly, then only you will get the results. That is very important. And the conclusion, and the best molecule, you should give a clear description. What is the exact SAR? Don't write don't write all the structural relationship. You please extract the best SAR. Okay, there are many structural relationship you found from the study. You please extract the best SAR, and you can conclude. Okay, these are the, I think for if a person want to publish paper, this is the most important. That is journal of ACS publication. These are the, I think I will give a slide for you. Then you can easily identify that whether it is very difficult, or difficult, medium, all the things I mentioned. But impact factor is not main. What is that? It is a 2018 impact factor. Now 2019 impact factor came that. But anyway, the competitiveness is same only. This is a Royal Society of Chemistry. What are the journals, best journals you can introduce your work? This is Elsevier publication. I didn't work on many journals, some main journals only. Okay, and please don't think that that Dr. Bijo gave this slide, so I can take the computational theoretical chemistry, the easiest one. I can choose like that. Don't think like that. You please try with the very difficult journal. Once you are trying the difficult journal, you will get the response from the reviewers. From there, you try for the difficult. When you try for the medium journal, and finally you can land the essay. You please always dream your project in a higher manner. Okay, this is the Wiley journals. This is a Bendam journals, okay, and this is Springer journals, okay. Okay, please don't publish these 1,000 rupees or online fake journals. I think tomorrow Dr. Karthikeyan sir will take a detailed about tomorrow. I will my topic is please unmute your mute your audio. Okay, I think tomorrow. Hello. Tomorrow we will see about if you have a dream in your molecule, if you have a dream on your molecule, what are the simplest software you can employ to design your molecule, and how to make the international collaboration, and all the things we will see finally. Along with that, the Dr. Karthikeyan will take that what are the what is that local or fake journals. So many peoples, I think, especially in Kerala, many peoples they are publishing their papers in fake journals, something like 1,000. If you're giving 2,000 or 3,000, you can see a number of journals. Okay, don't publish such type of substandard journals, so your work will not be recognized. If you want to recognize your work with high platform, with high international level, so you please okay, I forgot about the nature and other things. Okay, so first, if you are publishing, if you are satisfying this, then we will try for the nature and other big publishing, something like Oxford and Nature. Okay, so hope this is the last conclusion for this is the simple suggestion. I think this particular picture is very clear. Okay, so I think after this, inspired from these slides, I think someone's mind have a research curiosity to start a project. But you please note that, you see, Apple is just falling because due to the law of gravity. Don't think that, oh, I can make another thesis. If it is orange, if it will fall, that is also a law of gravity. So here the object is only changing, not the fundamental rule. So that is high impact paper. The next one is low impact factor paper. So these are the simplest demonstration. I can compare the low impact and high impact. So that's a final thing. You please always learning is a continuous process. Please stop that, and your learning is for your satisfaction, not for the position, nothing. Okay, please stay hungry, stay foolish. So that's all. If you have any doubt, you can ask.
Sir, please stop sharing your screen.
Okay, I think in last session, I want to explain more in the NMR spectroscopy. Okay, we will make another section for in these areas, because that are very interesting aspect of NMR and the C13. Okay, that we will see in the and docking also. I didn't explain much more. Okay, because due to the lack of time, I think hope you are getting bored. I think I took more time. Sorry for the trouble. Okay, I think we can
Sir,
stop
can
this share.
can I please stop sharing the screen, sir? Yeah, fine,
Okay,
fine,
okay.
fine. Now the session is open for discussion. Do you have any queries? Please unmute your video, introduce yourself, and you can ask the question. Now the session is open for discussion. Those who have any queries.
I think the participants are getting. I think they are not in a mood by hearing all these. I think I didn't tell much more of chemistry.
Yes, sir. There is a query, sir. Please introduce yourself. Unmute your video and introduce yourself. And please ask your queries or questions, whatever.
Sir, good morning, sir. Bijo, sir.
Hello, can you tell what is your name?
Sir, I'm myself Desna, working as an associate professor, JDT Islam College of Pharmacy, that is Kerala.
Okay, madam. Tell me.
Sir, I have a doubt in the introductory part. That is, you have mentioned some journals, no, sir.
Okay.
Sir, can you please repeat those things that we have to take care while writing the introduction? The important things, and regarding the journals also.
Journals, I gave the list because at the end of the session, the Karthikeyan sir will surely share your what is it, your PowerPoint. I will give you. You can see the list of journals. In the introduction, mainly you please target on the target of your disease. Pathogenesis target. Then you go for the chemistry of your molecule. Go for the previous history of the molecule. Go for the FDA approved drugs of the corresponding nucleus, whether it is included in the target or not. Then go for the SIR, find the gap, make the objective. That's all.
Okay, sir. Especially for SAR, can you suggest some journals? I have seen the list of journals, but especially for SAR, can you suggest?
SAR because medicinal chemistry, in all medicinal chemistry, SAR is accepted because without SAR we cannot publish a paper.
Yes,
SAR,
sir.
SAR means it is a structured history of your work, but you should interpret in a different way. Because you synthesize the molecules, you established a series of biological activities. Suppose a third person, if he's seeing, so he is identifying that maybe this type of changes will influence the activity. So you know that it is entirely a different perspective angle of a human being. That's why I told you that the observation is the most important thing. You please more read a research article, you will get a slight idea. No issue.
Okay, okay.
Any
Okay,
more
thank
doubt?
you, sir.
Okay, okay, madam.
Thank you. Any other queries, please?
Hello, sir.
Yeah,
Yes.
please
Good morning, sir.
unmute your video, madam. Please introduce yourself. Yeah, please.
Hello, sir.
Yeah.
Good morning, sir.
Okay, okay.
Sir, this is Chaitra from Mangalore, sir.
Okay, okay. Tell me.
Sir, in the purification of the compound, just washing with water and recrystallization is enough, sir.
For single step.
Because I had some problem when I was doing my PG, sir, in the purification.
Madam, yeah, because recrystallization, that's why I told you, know, in the side itself, I didn't go through the column chromatography. Suppose if your molecule